Hello. Thank you, everybody, for joining today. My name is Dr. Bogner. I am a functional medicine physician focusing specifically on autism, but also other chronic diseases. And this is my first broadcast here, trying to see if we can form a community to discuss autism. And I'm very interested in particular pathways in my practice to identify areas that we can help these children with. And I wanted to present a model today for autism and kind of go through to see what evidence there is to support these claims. And for the ultimate benefit of helping these children. I see a lot of children over the last 4 years, helping a lot of families and trying to make sense of where to start, what we can do to at least get some relief. And I have to say, over the last year, it's been an incredible journey with many of you that are listening here as well, from having children that have tried everything under the sun with their parents. And we have incredible new insights, in my opinion, over the last several years, gained more understanding of the abnormalities, the deficiencies, the toxicities, and kind of trying to present this in a way of understanding where do we start. And there is so much chatter about pandas, Lyme, autism, co-infections, deficiencies, metal toxicities, and what can we do? Where do we focus on? There's different groups that form all over the internet in regards to these subsets of things that we can do, but it seems that results are so, they vary. If we ask, hey, what works for your child with autism, we all know it doesn't apply to necessarily my child. And so I wanted to present a model today, kind of give an overview from 40,000 feet to see what's going on here. And again, what evidence do we have? So I don't know how long this is going to take, but just bear with me. If there are any questions anywhere, I hope I see them. I have the chat here, 2 comments already. Hi from Ireland. Well, that's nice. Hello. Let's see where everybody's from. But so far, there's not too many here. But anyway, let's get started. Hopefully, everybody's able to hear me clear and see everything well. So let's see how this goes. So, created a little bit of a chart here, picture that I've posted on my social media as well. So this is where we're at. And this is how I see the core issues with autism, where the core issue with autism lays. And it's, in my opinion, a self-sustaining system that feeds off itself in patients with autism. It's very complex, so it's kind of hard to start somewhere. But I think we all can agree that one of the major issues with autism occurs in the gut. And a lot of our kids with autism, I think it was up to 90% of children with autism have some sort of GI problem. And so let's start right there. So we see here that in the gastrointestinal system, if we have a problem such as, let's say, yeast, mold, bacterial overgrowth, Clostridia, all of that, we've seen this all in our testing with our children, that what happens is that the inflammation that occurs because of this overgrowth creates leaky gut. And leaky gut is not just a term that exists for nothing. I mean, it was discovered in the 1980s once they administered alcohol to rats, that that's when it first was coined, a leaky gut, that the GI system starts leaking things into the bloodstream. Well, into the bloodstream, into the portal vein that drains everything that's coming into the liver. And so usually most of the toxins that are contained in our food are passing through the GI system without being absorbed. However, if you have a leaky gut, as we all know, food protein, toxins, and also, as we will learn, the toxins from the mold will start to enter the portal vein. And what's interesting is that everything from our gut, in an adult we have about 20 feet of gut, everything first gets drained into the liver. The liver is our border control. It's a sponge for the toxins. The purpose of the liver is to prevent that any toxins leak into the blood. And I'm very sorry, but my dog is barking and he needs to go out, so I have to get up for a second. 1 second. So he's fine. And so we have our liver, which of course tries to prevent that any of those toxins leak into our bloodstream. And the way the liver does that is by grabbing the toxins, by binding them, and then neutralizing them without causing any drama, without causing inflammation. And that process is done usually with glutathione. Glutathione, 70% of it is produced in the gut and in the liver. And glutathione is our master detoxer, and it's also very, very important molecule for inflammation. And so we know from studies that children affected with autism have very low levels of glutathione, not only in the liver, but also in the brain. That will come in important in a little bit. But when we have an inability of binding chemicals, such as food proteins, toxins, then the next step in the liver will be to blow up these chemicals. And we do that, or the liver does that, with oxidation. Chemicals in the industry that are being used are usually very negatively charged, meaning they have a lot of electrons in them, and which makes them fat soluble. So, if we're depleted of glutathione because of production problems, because of binding problems, as we'll see in a little bit when we look at the genetics, then the body or the liver is trying to make these chemicals more positively charged, because the more positively they're charged, if we strip these chemicals of electrons, then that makes them more water soluble, so we can pee them out. But the problem is, if we have production problems with glutathione, or because of so many chemicals coming in, we're depleted of glutathione, then oxygen will react with the chemical, making them less negatively charged. We can pee them out, but that comes to the expense of having created free radicals. And a free radical is simply an oxygen molecule with an extra electron that it obtained from the chemical. And oxidative stress is a big problem. As we all know, specifically in autism. I mean, I remember Dr. Richard Fry from the University of Arizona, in one of his studies with Rossignol, found 78 studies that implicate oxidative stress with autism. So it's a thing. So and that's mostly because of the lack of glutathione again, and the leaky gut. So for one, we have produced a lot of oxidative stress, a lot of free radicals. But on the other side now, there are going to be a lot of chemicals and toxins and food protein that start, as you can see here, leak into our peripheral circulation. And I don't know if you see my mouse here. Once any chemical enters the peripheral circulation, and I always give that example. For example, a mosquito bites you in your hand. Obviously bypassing the liver, a toxin is injected into you. What happens? Well, immediately the body releases histamine. Histamine dilates the little capillaries around the toxin, so the immune system can gang up on that toxin that was just injected and prevent it from circulating in the periphery. And that's why we see redness, that's why we see itching, and that's driven by histamine. Now, what happens if there's a bigger toxin, and let's say you don't have good abilities of the immune system to cope with the toxin, like, for example, a bee sting. It's a little bit more serious toxin, gets injected, hurts like hell, and that usually gives us a bigger bump. But in certain individuals where the immune system is incapable of neutralizing that toxin, it starts traveling. And what happens is that a lot of histamine gets released along the way, which causes peripheral vasodilation, and in some individuals that are not so lucky, causes anaphylaxis, hypovolemic shock. And when you are one of those individuals and you go to the ER, what usually saves your life is adrenaline. You get an EpiPen and you're all set. The reason I mention that is because the same happens if the liver starts leaking chemicals into the peripheral circulation. When that happens, histamine gets released, as you can see here, right there. Histamine gets released, and histamine, consequentially, will trigger the release of adrenaline. And once we have adrenaline, a lot of things start happening, and that usually depends on the genetics of the individual. And so it's getting a little complicated with that, but as you can see, adrenaline has a lot of effects on us. For one, as we all know, it's the fight or flight molecule, neurotransmitter, that puts us on high alert, dilates our lungs to let more oxygen in. It recruits a lot of blood sugar in our skeletal muscle in order, to predict that we're going to run away or fight or flight. In particular, in individuals affected with autism, and I've been doing genetic testing and neurotransmitter testing for the last 4 years, you start seeing patterns. And you try to explain, why do certain individuals have high-functioning autism? Why do some individuals have low-functioning autism? And that's very, very complex. However, as a basis, we can see that there are problems with the breakdown of adrenaline. If you see a car accident and you're all shaken up and or something terrible happened, you can see that once you come down from that adrenaline rush, you're still shaking. And you give it another hour or two, the shaking has gone. And that's because there are enzymes involved that break down the adrenaline. And in certain individuals, those enzymes just don't get produced enough. And as you can see here, I'm trying to make this a little bit bigger. As you can see here, the principal gene that breaks down adrenaline is called COMT, catecholamine O-methyltransferase. And that gene I often see involved in autism, and there are studies that confirm that as well. And so if you have a problem with this gene, and on top of it, have problems with the cofactors, or vitamins, or minerals that stimulate that gene, in this case, it's methylation energy in the form of SAMe. Or, for example, you have high levels of serotonin, which I also see on the neurotransmitter testing, which further inhibits COMT. We have high levels of adrenaline. And what happens with adrenaline, especially under the influence of oxidative stress, adrenaline starts to oxidize into adrenochrome. And adrenochrome is very much involved in neuropsychiatric disease, such as schizophrenia. There's no direct studies in regards to autism. However, autism and schizophrenia were one and the same diagnosis until 1984, when they were separated. Why were they separated? I don't know. But according to the reasoning and in the psychiatric manual. I think at that point it was the DSM number 2. They said that children affected with autism do not have hallucinations like adults have with schizophrenia. But if you think about it, if the majority of these children affected with autism, if they cannot speak, how do we know that they're not hallucinating? On top of it, a lot of high-functioning autistic patients that I talk to, they do see things. They do sing, they do see things, and they hallucinate. But if you look at adrenochrome, at the history of adrenochrome, of the studies that we have, and there are a lot of studies with adrenochrome, especially when they administered adrenochrome to volunteers, you found a more depersonalization. Your reality literally shifts. You have more anxiety because of it, paranoia, like I said, hallucinations, which most of the hallucinations, 80% are auditory, the rest are visual, but then there are also taste and smell hallucinations that can occur with adrenochrome. And so that's one thing about the adrenochrome that's maybe not so flattering. However, adrenochrome is also antimitotic, so it's actually very effective against the development of cancer. In fact, schizophrenic patients, for example, despite, I think I read somewhere, up to 80% of them are smokers, the cancer risk is about 40 times less than the general population of schizophrenic patients. They live long lives. So, and that is because of the adrenochrome. And so that, in my opinion, with those individuals that have these issues with, these issues with the breakdown of adrenaline, they are high functioning, because the spatial awareness that adrenochrome produces is actually pretty fascinating. Of course, in higher levels, it's a problem, but adrenochrome, in my opinion, from what I find, gives you a better intuition. A better intuition, you can read people better. It's almost as if you have that sense that, when you walk in a room and you feel this negative energy, that sense is much more potentiated with adrenochrome. And I've talked to a lot of people, including adults, who have these genetic variations, and some of them are suffering from it. Why, for example, is a schizophrenic patient isolating from other people? It's not because they have a lack of confidence. It's because it's almost that they can feel the energy from the other person, and they don't want to absorb that energy, which is oftentimes a lot of negativity. You can see through somebody if they're being honest with you or not. And so those are the kind of things with adrenochrome. And that's why I see a lot of high-functioning individuals with autism. They're very intelligent, yet they usually have a more negative outlook on life and on themselves. Is because of those reasons. And so it's kind of a silver lining with adrenochrome, especially in children, I think, where the brain is still developing. It's very confusing. Now, on the other side, we have bufotenin. And bufotenin is a pretty incredible finding, I think. There are 4 studies, first of all, that showed that individuals with autism and psychiatric disease have elevated levels of bufotenin in their urine and in their stool. But what is bufotenin? Now, if you look at this here, if we look at the breakdown and metabolism of serotonin, usually serotonin gets broken down by MAO. It's a gene that produces an enzyme that breaks down serotonin. Very easy to understand. Now, what's interesting is that MAO is a gene that's only located on the X chromosome. It's not on the Y chromosome. So that means that boys only have 1 copy. Girls have 2 copies. And if we have this gene compromised, let's say particular patient, male patient, received a weaker copy of that gene and works slower, then we have higher levels of serotonin. And I mean, a lot of us have that. So what makes it different from normal development to autism is the fact that the MAO gene can be further inhibited by toxins such as aluminum. And there's a lot of aluminum around. Certain, I mean, it's all around us. It's the most ubiquitous metal on the Earth's crust. But it's also contained in a lot of other environmental treatments or prevention treatments, such as vaccinations. By the age of 18, I think child following the CDC schedule receives about 5,000 micrograms of aluminum, which has been shown in, I don't know, many studies to be neurotoxic. However, aluminum inhibits MAO, which then raises the serotonin. The serotonin then inhibits the ability to break down adrenaline, and the adrenaline will form into adrenochrome. But furthermore, what's very interesting is that now, it starts to back up. There are building blocks for serotonin, such as tryptamine. Tryptamine forms into 5-HTP, 5-hydroxytryptamine, into serotonin. But tryptamine starts to build up because it doesn't have anywhere to go, because you can't break it down. And I see this all the time. I've been doing neurotransmitter testing for a long time now, and it's very rare that I see a patient affected with autism with low levels of tryptamine or serotonin. They're always the most sky-high ones. And it also would explain why the ratio of autism in males to females is about 4-to-1. Schizophrenia, same thing, 4-to-1. Is there evidence that MAO is affected in autism? Oh, you betcha, there is. And so tryptamine starts to build up. And why is that important? It's because tryptamine is literally the building block for psychedelic chemicals to form. For example, LSD, psilocybin from magic mushrooms, NMDA, mescaline, and also DMT, dimethyltryptamine, are all very powerful psychedelics. They interact with the serotonin receptor 5-HT2A, which coincidentally is the one receptor in the brain that is being blocked by the only approved medication for autism, or one of the only approved medications for autism, which is risperidone. Risperidone blocks the 5-HT2A receptor. Why? Why did they target that receptor? Do they know? I don't know. But what's interesting is that risperidone, well, it's not interesting, but it sucks, because it would be great if risperidone only would target that receptor, but unfortunately, it targets about 15 other receptors. And so obviously we have side effects. I think about 80% of kids on risperidone gain weight. So it's not an optimal choice because of these side effects that occur because of blocking too many receptors. But anyway, so we have tryptamine now, and that's high. So what happens? Well, what happens is that tryptamine, when it receives methyl groups, specifically 2 methyl groups, it's forming into what we call dimethyltryptamine. As you can see here, the chemical name for bufotenin is 5-HODMT. 2 methyl groups, dimethyltryptamine. That is one of the core reasons why I do not use methyl groups in autism. I never had success with it. Very small subset of patients I do, especially if these genes work faster rather than slower. But not to get too confusing, I do not use methyl groups. I think the whole science about methylcobalamin, methyl B12 shots, it's not the right choice for these kids. No doubt they have methylation problems. But if you administer methyl groups to a child who has this going on, I don't think you have good results. Maybe for a day before these kids literally start tripping out into a different dimension. I mean, and it might sound funny, but bufotenin is one of the most powerful psychedelic substances known to man. In fact, it's used in shamanic rituals, not rituals, shamanic celebrations rather, that you can attend, where they mix DMT from a plant with another plant that contains a MAO inhibitor to make sure that the DMT is not broken down too quickly in the gut. They take DMT, they drink it. And what happens? Well, first you puke your brains out, but then after about an hour, you start to transcend. You leave your physical body. You see the matrix of the universe. The reports are pretty profound. The colors become brighter. Some say you can smell colors, you can taste colors. It's a different universe that you enter. And for some, it's actually soul cleansing. It's not dangerous, but for about 3 to 4 hours, once you drink an ayahuasca drink, that's how it's called, you are somewhere else. And so bufotenin, like I said, is elevated in the urine of patients affected with autism. One particular study, they had 46 individuals affected with autism and over 100 control subjects who were neurotypical. Every single child affected with autism had elevations in bufotenin. Only 2 out of the hundreds of controls had bufotenin elevations. So it's pretty significant. And there were 3 other studies. But if you think about it, that I label this here as low functioning, it's only low functioning to us, the observer, but they are in a different space, in a different reality. And that's why it's low functioning, because it's just for us, the observer, but it's just they're very detached from our reality. That's all I'm saying. And so there's a little bit of a difference for these 2 developments. Now, also very important to see here is that, why don't we have methylation energy to stimulate the COMT to break down, and why wouldn't we give methyl groups for this case? Well, for one, you can see methylation is formed ultimately all the way up here, where we see that we need to convert vitamin B9, from our diet, mostly coming from green leafy vegetables, or from dietary, fortified foods containing folic acid. But the body needs to convert all of that into the active form that we can use, called methyl B9. And there are many more genes that are involved in the conversion process of folic acid. One of them that we all know is MTHFR. Is autism connected to MTHFR? Well, you betcha, it is. And so we have less methyl B9 in a lot of these kids. On the other side, MTRR is activating B12. Where do we get B12 from? Well, red meat, fatty fish, highest in animal liver. And so B12 needs to be converted with MTRR. See, a lot of kids have these genes affected, so we have less methyl B12. But most interestingly, the gene that kind of fuses these together, even if we have methyl B9 and methyl B12, is called MTR. And MTR, which is also called methionine synthase, hold on, let me center this here, which is also called methionine synthase, which kind of fuses them all together to bring us this energy called SAMe that helps with the breakdown of adrenaline. That often I see affected as well. Usually to have 1 or 2 copies affected with MTR, it's about 3% of the population who have that affected. But if you do, and then you further inhibit it with aluminum. Acetaldehyde. Acetaldehyde is produced by yeast and mold. Mercury, lead, nitrous oxide. I'm writing a lot of letters to the hospitals for these kids, for our kids affected with autism who have problems with methylation, or going to the dentist where you get nitrous oxide. It inhibits MTR for up to 3 months. And so it has been associated with death. And so I write my letters to ask the anesthesiologist or the dentist if they can use a different approach, different gas, different substance other than nitrous oxide, and has worked. Some appreciate the heads up, some laugh at it, but oh well. Hydrogen peroxide. Hydrogen peroxide is a free radical that forms especially in the absence of glutathione. And so when we have that not working, because it's further paralyzed, we have less SAMe. Less SAMe, less COMT activation. I see it like COMT being your car, and the energy is the fuel for the car. And magnesium is the key for the car. So you need all of those to properly break it down to what it's supposed to be broken down, which is metanephrine. However, when that doesn't happen, adrenaline is high. We see a lot of hyperactivity. We see a lot of problems with sleep. Adrenaline induces, as we will see in a second, induces cortisol. Cortisol suppresses melatonin. Do we have problems with sleep in autism? Oh yeah, we do. A lot of problems with sleep. Everybody gives melatonin or hydroxyzine, or children wake up at 3 a.m, jump on the bed, they're ready to rock and roll. Not cool. So that's why we need to look at these. We need to look at these genes to see what we can do to at least mitigate some of these toxicities. Anything that accumulates and doesn't move along creates toxicities. So anyway, what was really interesting is then to see that the adrenaline starts to be chronically elevated. And we know that adrenaline ultimately, is our fight or flight response. If that's chronically elevated, it raises cortisol. Do we have evidence of higher cortisol in autism? Yes, we do. We're going to look at that in a second. But why is that important? It's because cortisol, when elevated, doesn't only suppress melatonin, it gives us sleep problems, because usually cortisol is very high in the morning, goes down throughout the day. But if we have high adrenaline levels, it keeps the cortisol up. And so cortisol is a steroid hormone. And if you know anybody with a chronic autoimmune disease that is on steroids chronically, you know that these individuals are more prone for yeast infections. To give you an example, I used to deliver thousands of babies in the hospital, and sometimes babies are born very prematurely. Mom goes into preterm labor. The baby delivers at 28 weeks, tiny little baby. The first thing that we do after we stabilize the baby is to make sure to address the lungs, because that's the biggest issue. The lungs are not developed. The child would suffocate if we wouldn't help it at 28 weeks. And so what do? We know, and that's common practice in the OBGYN world, in pediatric neonatologist world, to administer betamethasone, which is a steroid. We inject it because it has been shown to accelerate lung maturity by inducing type 2 pneumocytes, which are macrophages, to keep the alveoli open, prevents it from collapsing. But the reason I mention this is because when you now have administered your 2 doses or your betamethasone to accelerate the lung development in this child, and you come back 2 days later, oftentimes we see that these little babies have a white tongue all of a sudden, and that is candida. So I mention this because you see the powerful effects of steroids on inducing yeast overgrowth. And the same is true with anybody on chronic steroids. You're more prone for yeast, and ultimately, as you will see, mold overgrowth. And the reason for that is that steroids, such as cortisol, suppress CD4 and CD8 T cells. And CD4 and CD8 T cells are solely responsible, well, they have a lot of functions, but they're responsible for taking care of yeast and mold. And so, if you think about it, we have somebody with rheumatoid arthritis, let's say, and the immune system is attacking the joints. And this recruits a lot of fluid, and so that your hand hurts, it's swollen, you can barely close it, you can barely pick something up. It's because the immune system is attacking your joints. You give that person a shot of steroids, you tell these immune cells to go take a hike, you inactivate them. Swelling goes down and you can move your hand. Well, that's great, but the problem is that you also immunosuppress this patient. And now they're more prone for all kinds of things, including not being able to get rid of mold and yeast. And so we see that chronic stress causing elevations in cortisol, which suppress the immune system, and then that keeps the mold growing. And as we will see in a second, God, is there a lot of evidence that mold is involved in autism? It's pretty fascinating. And I'm so excited to present this because for the first time, really, in all of these years that I've been looking at this, we have some amazing results. I mean, and the results are progressively, getting better as treatment progresses. But we see children start speaking again. I have parents sent me videos of nonverbal children starting to sing in the shower, sleep better, have more attention, better reports from school, from the teachers. It's amazing. And even if it's a little bit that we help, for example, with sleep or with attention, it's huge and huge impact for families. I think only somebody who has a child affected with autism would understand how powerful just little changes can be. But obviously we're in the quest to eventually, have these children lose their diagnosis. And that might be an unrealistic goal, but why not? Why not have high goals to try and keep researching, and find, even if there's a small chance to find the best hope for these children, for our children, let them live, to their genetic potential. So anyway, so we have these mold molecules involved here again, and that keeps growing in the gut, causing more leaky gut. More leaky gut, stressing out the liver more depletion of glutathione. More depletion of glutathione means more toxins entering the bloodstream, which means more mast cell activation, which means more adrenaline. And ultimately, we'll see what happens in the brain here. Specifically, I'm very interested in this area called the amygdala. And the amygdala, probably talk more about it in a second. The amygdala is very much implicated in autism. It's a neuronal hub that receives input and sends out input. It's involved in memory formation, detection of threat, socialization, reading of facial expressions, and that is very much involved. And I came to the conclusion that, most likely what hyperactivates the amygdala is the toxins that are secreted by yeast and mold. And so that is huge. That is huge. That would explain everything, in my opinion. Why do these kids get better with therapy, with antifungal therapy, with binding therapies, with strengthening the liver? And also, why does suramin work? Suramin deactivates the cell danger response. You think about that. It deactivates the cell danger response. So even though suramin most likely tells these neurons in the amygdala that, hey, There are no toxins around. Chill out. That's how suramin works. But it didn't take care of the toxins. And that's why I think that Dr. Navio is researching this in San Diego, having amazing results because the amygdala is calming down, but it didn't take care of the mold. And that's why a lot of these kids needed another suramin infusion after 6 months. So with that being said, let's see how we can support this hypothesis with some awesome research. So bear with me. Let me open another window here. Where is it? Uh-oh. There we go. All right. So keeping this in mind here, our pathways. Let's look at this. So, and oh my God, before we get into this research, let me see if there are any questions here that I see. Hello from Turkey. Hello, I had a lot of Turkish friends growing up in Germany. Wonderful people. I've been to Turkey. I've been to Antalya. Great food. Hello, I've done genetic testing for my 5-year-old son and he has autism. And the genetic test, it found sequence variant of uncertain significance. Those genes are, woo. I'm just wondering whether you know anything about these genes causing autism. Can you refer? So these two genes that, thanks for your question. These genes that were identified were done by a whole exome sequencing, it seems. You know, and these tests are a little bit different of the genetic testing that I do. These sequencing tests detect mostly serious mutations or deletions in genes. And that has never been really reproducible to find a common gene missing or mutated that are responsible for autism. So I do not believe that autism in that sense is a genetic disease. I believe autism is an environmental disease. And so I can't tell you about these genes that you mentioned here. Do they maybe contribute to the development? Certainly. But as we will see, every gene kind of contributes to it. That's why we see such a big spectrum. If we look at this graph here again, you know, what I'm looking at is to see if genes are simply working slower. And we all have these variations in our genes. Some genes are working slower, some work normal, some work even faster. You know, but you see here, for example, the genes involved that I mentioned here in the GI tract are, you know, FOT2, CBS, SUOX. You know, to give you an idea, for example, on SUOX, this gene in the gut. Why is that important? Well, it's called sulfur oxidase. And what happens in the gut, you know, why is sulfur so good? Well, in some kids with autism, it's not so good. But why is it so good? Because sulfur, you know, in foods like cabbage, eggs, garlic, broccoli, you know, the cruciferous vegetables, or even, you know, glutathione is sulfated. But all of these food groups are so good, not only because they donate to our body cysteine, which is an essential building block of glutathione. As you know, there's cysteine, glycine, and glutamate, but only cysteine is what's really required in our diet because we can't synthesize it. We can change other molecules around to produce the other two building blocks, glycine and glutamate, but cysteine is required in our diet. And so when we get in contact with cysteine, first of all, we store cysteine in homocysteine. You know, so when we don't eat anything, let's say we fast for whatever reason, religious or not, it's that we don't die of inflammation because we can't produce glutathione. So homocysteine is our reservoir for cysteine, and it's used in methylation processes, but also we can tap back into it to release the cysteine, to build glutathione when there is, for example, the trigger of free radicals free radicals trigger the formation of more glutathione production. That's how intelligent our body is. But in order to do so, to tap into the homocysteine, we need CBS. And we check that gene, I check that gene in the genetic test to see if it's functional. And that's why the genetic test is so helpful, to see if you have that affected. And also, what stimulates this gene to function in the first place, which is vitamin B6, pyridoxine, but more specifically, the active form of B6. And so what does that mean? Well, if we consume, for example, your generic multivitamin from Costco, it has pyridoxine hydrochloride in there. Well, that's not biologically active until a gene activates that B6 into the active form, which is called pyridoxine 5-phosphate, P5P. And so that's what the genetic test tests as well, and sees if we have maybe a slower CBS and we have a deficiency in B6, which, by the way, oftentimes you can see that on the OAT test. I think it's marker 52, if I'm not mistaken, but a lot of kids affected with autism have deficiency in B6. And so you produce less cysteine, you drain homocysteine less, which means ultimately less glutathione. Great. But in regards to SUOX, when you have variations in SUOX in the gut, then, what is the function? SUOX breaks down sulfur. And that's a good thing. That's why sulfur is good. Because when you break down sulfur with SUOX, a byproduct of that reaction is the formation of hydrogen peroxide. And we said earlier, hydrogen peroxide is a free radical. It is. But you want to produce that in the gut because it kills viruses, it kills bad bacterial overgrowth, yeast, mold. It's sterilizing the gut, basically, prevents overgrowth. And if you have a problem with that gene, which, by the way, is stimulated by molybdenum and thiamine, but if you have that problem, then sulfur starts to accumulate. And with sulfur, that needs to break down in a different pathway, and it produces ammonia and other toxins. So that's why these genetic tests are so important. That's why each child is different, has different issues that need to be addressed. But anyway, so in the gut, those things are going on that we can identify with the genetic test. If we go here to the liver, for example, there are the genes of GPX, GSDP, CAT, and CBS. And it might sound intimidating, but it's really not, because these are the genes that are responsible for binding chemicals, such as the GSDP. But the GPX, CAT, and CAT gene, and I forgot the SOD genes to put in there, they reduce free radicals. And from what I see, testing children on the genetics, probably, I would say 100% of them have the SOD genes that reduce free radicals involved. That's a story by itself, but it's pretty amazing. And there are studies out there that confirm that the superoxide dismutase genes, the SOD genes, are affected in autism, meaning, we're affected with higher levels of oxidative stress. And, then we go to the peripheral circulation, where we have our food sensitivities pop up. Do I test for food sensitivities? I don't test for them anymore, because if you have leaky gut, you're gonna have food sensitivities. And I have not found good clinical results by eliminating cucumbers or, very rare foods that are potentially good, because it just doesn't make sense to me. Why do you have food sensitivities? Why are these kids have sensitivities to basically planet Earth, to everything? It's because they have a leaky gut, because these food proteins enter the peripheral circulation and the immune system cross-reacts with them. I just tell everybody across the board, don't consume dairy, don't consume gluten. And the reason is that gluten from wheat and casein from milk, they're just very inflammatory. Why? Because structurally, they have sequences in them that look structurally similar to some of our protein that we have in our body. And so the immune system can eventually start cross-reacting and cause autoimmune issues. And that's where a lot of these pandas subgroups start to emerge. And, also, so autoimmune reactions, that's what I want to say. The mast cells, as we will see in the studies, very much involved. Histamine is a huge topic in autism. Why is histamine high? Well, it depends on the individual, because if we look at a mast cell, in the peripheral circulation, a mast cell contains not only histamine, but also cytokines. And so specifically in regards to histamine, there are two genes responsible primarily for histamine. So one is called DAO, diamine oxidase, and the other one is HNMT. And the difference is that the DAO gene is breaking down histamine within the mast cell, and the HNMT is breaking down histamine once it's released. And so why does that matter? Well, if you have a problem with DAO, then that simply means that your mast cells are packed with histamine. And the moment that your mast cell gets in contact with an antigen, a mycotoxin, a food protein, any antigen really, histamine gets released. And then you have massive release of that histamine because you couldn't break it down within the cell. And then depending on if you have the HNMT involved, well, then depends on how quickly you can break down that histamine. Obviously, if you have big issues with HNMT, then histamine sticks around longer in your plasma, constantly, stimulating the release of adrenaline. And so that's how we can kind of piece it together one by one with the genetic involvement, and a lot that we can do about it. But anyway, to get back to the studies that I wanted to mention, keeping our picture here in mind, I wanted to start with this from my friend Russell Blaylock. As many of you know, he's at University of Louisiana. He's not really my friend, but I want to be his friend. But he's a good friend in spirit. He's done a lot of great work for autism, and I wanted to start with this because he came to the conclusion after all of his years of research that there is chronic microglial activation in the autistic brain, basically saying their brains are inflamed. And he published this in 2009. Multiple other papers, of course, confirming that we're dealing with neuroinflammation in autism. But the reason I mentioned this is because he relates this activation of these microglial cells to be in an inflammatory state because of the environment, not because of inborn genetic defect of some sort. He concludes it's the environment. So it's the epigenetics, not the genetics. It's the environment. So moving along, a study from Toronto, and again, like I said, there are many studies. I chose those. I don't know why, but they're good studies. This one was the one that showing that environmental factors already prenatally affect the brains in autism. Here saying that this is the first comprehensive summary of environmental factors such as exposures to chemicals in air pollution, pesticides, and consumer products ultimately lead to inflammation. What exactly? We don't know. But like we said here in this example, or picture, I make the case that it's mold and yeast that does that. So let's look into that. Maybe that's a little bit small. Let's make this bigger. I hope everybody can see this. So in the year 2000, William Shaw, who is the CEO of Great Plains Laboratory, he conducted a study, a first of its kind, where he treated children affected with autism with the antifungal nystatin. And what he used is 100,000 units 4 times a day for 10 days. And lo and behold, what did he find? That the children improved significantly. And he concluded that antifungal drug therapy might be a promising method for the treatment of autism. And so that was kind of the pilot thing that got that started. Now let's take a look at how these toxins from mold, what effect they have. Now, what's important is to realize, why does mold, the mold cells by themselves, why do they release toxins? What's the purpose? And if you think about it, if in nature, we have over 200 species of mold that have been identified around the planet, and probably in the deep end somewhere in the world, there are probably mold species that haven't been discovered yet. But the ones that we know of, they always fight with each other. So if one mold starts to attack another species of mold, the one that's attacked will release toxins to basically tell the attacking mold to back off. And that's why there are different toxins, because different toxins, are basically a defense method of the mold. And so when mold starts to colonize us in the gut, for example, it usually releases toxin, but it especially releases toxins when the mold is under threat. It's a pretty intelligent organism, if you think about it, because as we will see, eventually those mold toxins will make it to the brain. And as you can see in this picture, well, they have a long way to go. They have to basically overwhelm the liver, in the blood, have to overwhelm the immune system, and then eventually, make it to the brain. And so that's a pretty tough task for these toxins to do that, but it does. And eventually, as you can see, there's a phenomenon that occurs because of that called the amygdala hijack. It's a pretty dramatic term, but it's a real thing. Amygdala hijack. Question is, who's hijacking our amygdala? And I tell you, it is the mold toxins that do that. So let's see. How does the mycotoxins work? What do they do? First of all, they're unlike bacterial toxins. They have a different structure. They basically produce, will lead to the production of cytokines. Because as we said, any antigen that gets in contact with a mast cell activates the mast cell to release histamine, to dilate the vessels, recruit the immune system. It's an alert system, but also releases cytokines. And what's interesting is that cytokines have been involved specifically in other studies, and I'm not sure if I'm going to have them here or not. Cytokines weaken the blood-brain barrier. So that's one way how these mold toxins start to evade our immune system and make their way to the brain. So that has been validated. Ochratoxin A is probably the most common toxin that we find when we do mold testing. Ochratoxin A is produced by Aspergillus, by certain types of mold, but also by yeast. And so, ochratoxin A reduces mitochondrial function, apoptosis in neurons. But as we can see here, in mice studies, activates microglial cells, which would support Blaylock's conclusion, and multiple other studies that show microglial activation in the brain. Microglial cells, remember, microglial cells are truly the intelligent cell in the brain. If you have two neurons in the brain, they don't see each other. What connects them is the microglial cell in between, because the microglial cell has about 1,000 arms And it not only senses the environment for a threat, but it also takes these synapses of the neurons and connects them. So the truly intelligent cell is the microglial cell. The problem is, while the microglial cell wants to always connect and disconnect neurons to form memory and to kind of learn from our experiences in life, if one of these arms from the microglial cell senses a foreign antigen, such as from a virus, from a bacteria, or for example, a metal like aluminum, it starts to immediately sense a threat, and it's simply transforming into what we call activated state. So it's in kind of an alerted state where it stops connecting neurons and sends out more cytokines to attract a very specific immune response to take care of whatever activated it. A virus will recruit different immune response than a bacteria, and a different immune response than a toxin, for example. And so that's the function of a microglial cell. Truly amazing cells, but if they're chronically activated, we don't learn really a lot. We have brain fog, we have memory problems because we cannot form new synaptic connections. Very interestingly is that mycotoxins also disturb the olfactory bulb. And that's very interesting because not only do we see that, the olfactory bulb is our olfactory nerve, one of the nerves along with the vagus nerve that pierces through the skull into our nasal cavity and has its little projections in the nasal cavity. And so we smell things. And, it's one of our senses. And we see that in autism. I mean, they have all kind of aversions to foods. A lot of kids come to you and they smell your hair. They like different smells, so it's disturbed. But the reason I mentioned that is because what's very interesting is that the olfactory nerve, when it goes to the olfactory bulb in the brain, is literally 2 synapses away from the amygdala. And so when we will realize later that mold also tends to colonize in the sinuses, and the mold toxins released in these sinuses could potentially activate the olfactory nerve to directly trigger the amygdala to be hyperactive. That's pretty significant. But also here we can see that mycotoxins have been involved in encephalitis, brain inflammation. Here's a great chart from the company that I do mold testing with, from Real-Time Laboratories. I do not use Great Plains anymore. The reason is that Great Plains just takes too long. I don't like to wait that long. They take up to 8 weeks to have the results come back. I recently switched to Real-Time. Not only do they check for more toxins of mold, but the results come back within 7 to 10 days. So as we can see here, it's probably a little bit small. Oh boy. I don't know how to make this bigger. Oh, here. Oh, it doesn't go bigger. So anyway, this simply gives us an overview of the toxins that are being checked with this test and the effects that these toxins have on our system. So for example, this one causes immunosuppression. This one inhibits mitochondrial energy production, stimulates inflammation. Again, immunosuppression, inflammation in the nasal cavity, mucus, olfactory system, inhibits protein synthesis. Interestingly here, the last one, the zearalenone, is an estrogen mimic. So it can lead to reproductive issues, low sperm counts, spontaneous abortions, early puberty in girls. So anyway, it literally can impact every cell in your body. And the interesting thing about mycotoxins is that they can spread from cell to cell without entering the plasma. So it's a very intelligent organism of how that works. And It also takes some time to treat because it's trying to hide. But anyway, how are mycotoxins involved in autism? So here's a pilot study out of Italy, and it showed that when they analyzed 52 autistic children, they were compared to healthy children, and they found a significant association between ochratoxin A in the urine and the blood in children with autism. That was statistically significant. Even though it was a small study, it's pretty interesting. This study also comes from Italy, and what it showed, it looked at 172 kids with autism. It had 61 controls, and showed statistically significant differences comparing mycotoxins, again, with the children who were affected compared to those who weren't, in different types of toxins. So in this study here, we see that ochratoxin A can generate neuroinflammation by, again, activating microglial cells. And we do have microglial hyperactivation in the brains of autism. A lot of studies out there. This one was interesting. It compared neurobehavioral and pulmonary functioning between mold-exposed boys with autism, non-mold-exposed boys with autism, terbutaline-exposed children, and unaffected children from a community with no known chemical exposures. And they compared all of those and found that mold-exposed boys with autism averaged significantly more abnormalities than did the other groups, especially in balanced vision and blink reflex latency. And so if we move up the ladder here on our picture, and we basically start to focus on the liver in relation to mycotoxins. Mycotoxin-related compromise of glutathione production can result in an excess of oxidative stress that leads to tissue damage and systemic illness. And so not only does it drain our glutathione, but it also hinders its production. And you can see that if you do have genetic predispositions where you're already making too little glutathione, that those are the individuals that will suffer, suffer the consequences, and will move on to the next step more excessively. This study was done by Dr. Adams, James Adams, who's doing a lot of work in autism, specifically relating to the gut now. But back when he brought out this study here, he basically showed that there were statistically significant differences in the ratio of available glutathione to oxidized glutathione, and found that in autism we have these children affected with very low levels of reduced glutathione, which is the active form of glutathione. And consequence, as we said, is a lot of formations of free radicals. Here are studies that show that there's oxidative stress due to the lack, most likely because of glutathione. What areas are affected in autism? Interestingly, as you can see here in the study in 2008, the area that's affected is BA22, which is Brodmann area 22, which is the area responsible for speech. And you see a lot of different areas in the brain can be affected, but you can see the just sheer volume of information that we have just in the ASD brain that glutathione is lacking. Why? Again, leaky gut, toxins, genetic inability to really produce a lot, to bind to toxins, So our glutathione quickly gets drained. Again, if any of you have a oat test, look at the marker down at the bottom, somewhere in the 50s range. In regards to NAC, I bet you it's 0. And remember, NAC is cysteine, the building block for glutathione. Why is it 0? Because it's being used, not because it's not consumed. Furthermore, Dr. James pointed out that Brodmann area 22 in the region of the human brain, again, responsible for speech, is responsible for the generation and understanding of individual words. And the levels of glutathione were significantly decreased in the cerebellum and Brodmann area 22, causing this area to be inflamed. So that's why we see a lot of speech problems. So if we move up now to the histamine, we have a study here, kind of just shows that histamine release leads to an excess of adrenaline. Yeah, this one is just specifically to relating to the mast cells, that contain the histamine. And I found this very interesting that a lot of studies have shown a strong correlation between autism and maternal or infantile atopic diseases such as asthma, eczema, food allergies, food intolerances, all of which involve the activation of mast cells. So these new tissue immune cells are located perivascularly in all tissues, including the thalamus and hypothalamus, which regulate emotions. Mast cells derived inflammatory and vasoactive mediators increase the blood-brain barrier permeability. That's why I wanted to bring it out here, because if the mold activates the mast cell, it literally creates its own path to get access to the brain by the release of mast cell cytokines. And we have some of these here: interleukin 6, interleukin 17, tumor necrosis factor. And so toxins then have the access to the brain. So let's see what happens. And specifically, I'm interested in the amygdala in autism. What happens? So the amygdala, just some facts here. Now, each one is from a different study. But the amygdala in general detects threats. It's important for attention. It decodes facial expressions. Often, if the amygdala is hyperactivated, the child looks you in the eyes, it detects a threat. That's why they have poor eye contact. They have found in studies that if you make these children look you in the eyes, that with studies that the amygdala starts to boot up, it starts to be hyperactivated. They see it as a threat. The amygdala is a key player in social behavior, how we integrate our surroundings of what's happening around us into how do we process that, behaviorally. It's involved in schizophrenia, anxiety disorders, bipolar disorders, and in autism. We know that in children affected with autism that the amygdala grows faster. In my opinion, the only reason is because it's being permanently activated. In autism, the amygdala has more neurons. It is basically, amygdala means almond, and it's in the shape of an almond. But it's such a complex structure, even though it's so small, because it receives input from a whole different areas of the brain. It connects to a lot of different networks in the brain. So one of them is the hippocampus, which is responsible for translating your experience into memory. And it also links to the prefrontal cortex, which modulates and controls your emotional responses to your experience of life. Also links to the BA22 area that we just mentioned, which is compromised in children with autism. And so, that's a big thing because, we see in these newer techniques, or newer to me at least, techniques of spelling to communicate. We teach these children how to point to certain letters to form sentences. And my God, have I seen amazing videos that showed that even children that you would never think that they could even form a sentence, they write paragraphs. And I've seen parents send those to me, and children with autism who cannot speak and behave violently at times, apologizing through a system like this, that they behave this way. They are apologizing because they can't help themselves. Think about that they know what they want to say, but they cannot, because the connection between Brodmann area 22 and the amygdala is not working. Because the amygdala receives input from Brodmann area 22. And what it does, it translates your intent to talk, that signal, into motor neuron activity into your mouth to make it move and produce sound. But if the amygdala is hyperactivated, it doesn't work. So they know what they want to say, and they just can't do it. They have also found that the weaker the link between those different networks and the amygdala, the more severe, obviously, the symptoms of autism. The study from London showed, basically, they tried to visualize the volume of the amygdala in autism, and found there were increased volumes in the certain regions of the amygdala. The amygdala has many different regions, and so it's still very complex to understand, and probably different regions will create different varieties of autism on the spectrum. And so that is still, hot research topic currently. The involvement of the amygdala in memory storage. Here, I brought this study up is because the amygdala, let me just read that. The infusion of if you, for example, have beta blockers that block the effects of adrenaline, then we should have, in general, we should have a good response, because what they're saying is that adrenaline and steroids modulate the amygdala, so they hyperactivate the amygdala. And so I thought that was really important to mention, because we mentioned adrenaline here. It's fight or flight, so obviously adrenaline activates it, puts us in a higher alert state. This study was very interesting because it actually looked at the amygdala post-mortem in children affected with autism. And they had 30 post-mortem cases that with patients that had symptoms of autism, and they found increased cell packing density predominantly in the cortical, medial, and central nuclei of the amygdala. So again, why do you have increased cell packing there? It's because it's hyperactivated. This is a very, very good text that I found on this website here that I put together and put on my Facebook as well. But I think it's so amazing to realize what this effect of the amygdala hijack really is. Who's hijacking it? Nobody said, nobody points out to that, but let's read it. So the amygdala can be stimulated when faced with a perceived threat. If in a life-threatening situation, the amygdala will send information to other parts of the brain to prepare the body to either face the situation or to get away from it. This fight or flight response is triggered by emotions of fear, anxiety, aggression, and anger. It is beneficial that the amygdala is working correctly in order to act appropriately in threatening or stressful situations. However, sometimes the amygdala can act too strongly, leading to amygdala hijacking. Typically, in a stressful situation, the frontal lobes will step in to override the amygdala to ensure we respond in a rational manner. But if the stressful situation causes strong feelings of anxiety, anger, aggression, or fear, this can result in illogical and irrational overreactive behaviors to be displayed. Essentially, the amygdala overrides the frontal lobes to hijack control of the stress response. That's big, because in us as an adult, it has a different effect, obviously, than children, because their frontal lobes are not fully developed yet. For us, if somebody makes me mad, or something makes me mad, instead of violently bursting out and hitting somebody or screaming something in profanity, my frontal lobes tell me not to do that. But in children, it's just a struggle. Their brains are still developing, so it's obviously causing big behavioral problems. Now, this one is a very interesting study, because here we see, well, first of all, we know that there are certain molecules that fire up the brain, such as dopamine, for example, fires up our brain, gives us a lot of brain power, imagination, creativity, and so forth. Everybody wants dopamine. But there are also molecules, neurotransmitters, that have the opposite effect, that are what we call inhibitory neurotransmitters. One of them is GABA. And GABA slows down and cools down the brain. A lot of people take GABA when they go to sleep because they don't want to think about too many things that keeps them awake. And so GABA receptors are present on the amygdala. Now let's see what's going on here with this yeast and autism. So what they're proposing here is that yeast, Candida, produces propionic acid and ammonia in the GI tract. Then all of this stuff reacts and ultimately results in the production of beta-alanine, which is structurally similar to GABA and has been shown to be in higher quantities in autism. Well, that would be a good thing, but it's really not, because what it does, it acts as an antagonist. So even though it looks structurally similar to GABA, when it sits on the receptor, it doesn't activate it. In fact, it just sits there and prevents that more GABA can calm the brain down. So, which essentially means it's kind of like the blueprints of this amygdala hijack that, Candida or, Aspergillus produces that too, that these chemicals inhibit the green button on the amygdala that tells it to calm down. I think that's pretty profound. Here, if we move along on the picture with the cortisol, it's just a study that simply shows that, patients affected with autism had higher evening cortisol levels than the control group. And that's it. And so that's all I wanted to tell you. I didn't know it was the end already, but I hope that made sense. I hope that we can discuss this. So let's see. Let me see some of your questions. Please talk about MTHFR. I'm trying to address as many as possible. It's not too many questions. So can I talk about MTHFR? Yeah. MTHFR is just one gene that produces an enzyme that activates folate into the active form methylfolate, which is responsible for providing one of the building blocks to produce methionine, which is methylation energy. Over 300 processes in the body require methionine. Some of them include cell repair, muscle health, glutathione production, breakdown of neurotransmitters. So MTHFR is just one of many. I sometimes see children affected with autism that don't have any MTHFR involvement. And so It's more to look at the global picture, the genes that are before MTHFR, MTHFD1, FOLR2, DHFR, and the genes that follow that. So it's just one little gene. And I don't think with just checking MTHFR, you don't have any further answers and what to do. I think you need to look at the other genes, look at the interactions, and look how it all connects, and obviously see what symptoms you're dealing with. You can get a genetic test which looks horrible, but the person is super healthy. It's because they take good care of themselves. It's not a death sentence if you have bad genes. You can nourish them with the right nutrients and circumvent a lot of these, tendencies to create toxicity. So I hope that was a good answer. Thank you for stating this regarding aluminum. Yeah, aluminum. Let's see. What test shows bufotenin? I wish I had a test. I've been communicating a lot with researchers from University of Michigan. University of Michigan is one of those universities that does a lot of research on psychedelics, and they have some excellent researchers. One unfortunately just left that I was in contact with. He left, I think, to Colorado. But there's no test, unfortunately, for bufotenin. It would have to be by gas chromatography. Would need to be a study, basically. There's no commercially available test for that, unfortunately. Let's see. How long does it take to get rid of mold? Well, everyone is different. And, there's a difference between having mold just sitting there and to have a colonization of mold. And that, a lot of research, if anybody wants to look into that, the best research comes from Dr. Neil Nathan. He's a veteran in treating mold. He has written a book called Toxic. I highly recommend everybody reading this book. It's fascinating. And he basically mentions that there's colonization and there's just mold. And so a lot of mold you can take care of with just binders if there's no colonization yet. The problem is that if mold starts to stick around, it integrates into our systems a little bit deeper and creates a protective biofilm around it, which is usually mucus-based, and the mold is hiding in there, releasing its toxins and making it very hard to access by our immune system or medications. And so mold is not something like a strep throat. You can throw in some antibiotics, 5 days, you're done with it. No, mold takes some time, depending on how much you're affected, how compromised your immune system is, and if it's in your sinuses already or not. It's a tough one. It's an intelligent organism for sure. Let's see, what else? Croatia, beautiful country. Any thoughts on elevated levels of absolute CD8 and natural killer cells? Is this a good thing, or could it cause a problem as it relates to yeast and mold? Very interesting questions. I would have to look into that one. I'm not that smart. Hello, Kimberly Mitchell-Reings. Hello, you're listening. Wow. Is it possible to do the mycotoxin test through real-time labs in Turkey or if in UK or Europe? Not that I know of. So for those that are in Europe or in Asia or in Australia, I highly recommend, for those that are in Europe, to go to this website called biolab.co.uk. And they have all sorts of tests. You can do neurotransmitter testing from doctor's data. You can do all tests from Great Plains Laboratory, and they ship it to you anywhere in Europe and analyze that for you over there in the UK. So that's one way to do it. If you truly want the real-time lab, that can be arranged, but the ultimate thing is get the Great Plains lab. I don't have a problem with that test from Great Plains. It's just it takes so long. But if that's the only option, of course I would do it. But the oats test can also suggest the first 10 markers on the organic acid test from Great Plains also suggest if there are yeast and fungal markers. But the mycotoxin test is much more specific to very specific mycotoxins. So, but if everything fails and you have an OAT test, it can already indicate if there's something. Let's see. If both a metal level and mycotoxins are positive simultaneously, which should be addressed first? Well, if you think about that, the mold creates the access into our system for other toxins to come in, and draining the liver of its glutathione, I think it makes more sense to address the mold first, because if you close the leaky gut and prevent that more can stress out our livers, the liver can take a breather, produce more glutathione, and then start sucking out the toxins from our system. But the body is very intelligent. The body is always trying to make sure that the damage that has been done is not getting worse. So it always puts its focus on defending further damage. So the primary focus is always first to make sure that we take care of what's coming in is not making it worse. And so we have to stop that, leaky gut, whatever causes it, bacterial overgrowth, mold, yeast, and then let the liver do its job once the glutathione starts coming back up. Should we supplement with GABA? Good question. I had that question come up 2 days ago from a patient. Well, first of all, we can see that a lot of these toxins block the receptors, working as an antagonist in the brain. And so in addition, so it wouldn't really work. And in addition, there's a study, and I thought I had it here, but I didn't post it. But GABA is not very good at crossing the blood-brain barrier when you ingest it orally. And thirdly, if you have done a neurotransmitter test before, you can see that virtually everybody who gets tested, or at least most of the children with autism that I test, already have high levels of GABA. GABA is produced in the gut in the first place. But it has poor absorption into the brain. And even if so, again, the GABA receptors are already occupied by these other toxins that block it from being activated. So it's pretty dramatic. Let's see. How do you determine mold involvement and what treatment is available to reduce its impact? I think we answered that, with the testing of mold, can detect mold toxins in the urine. That's how we do it. And they have reference values, of course, when we compare mold toxins in the urine to neurotypicals, and that's how they come up with reference ranges. And, usually if you cross a certain threshold of the neurotypicals, then it's deemed to make a significant difference, and that you have that exposure to that mold. What's interesting though that I've found is that when a mold test comes back, and let's say you have 2 markers elevated, and then you start to initiate therapy, and then after a while you do another mold test, all of a sudden you have 6 markers elevated. How's that possible? They have all of a sudden new mold toxins that were detected in the urine. Well, if you think about it, the amount of toxin that is measured is from the urine. So that means that something is already fighting the mold inside of your system. So just because you have a negative, like let's say one of the mold toxins is not showing up, doesn't mean you don't have it. It's just it wasn't detected in the urine, which could mean that it still sits in your body somewhere, and unless you provoke it, it's coming out. And so that's why we see oftentimes, initiate therapy, for example, somebody's initiating therapy with antifungals, and then they do another mold test a month later or 2 months later, and they see much higher values than before. Well, that's a good sign. It's because your body's getting rid of the toxins. You only have so many, and the more you pee out, sweat out, and poop out, the better. So a couple more questions. Let's see. Oh man, a lot of good questions. Let me just pick the easy ones. Some of you are really smart, and I apologize if I don't answer all of them. Yeah, do you recommend glutathione supplementation? Is it possible symptoms might worsen before they improve with such supplementation? Yeah. Do not, and I just mirror what Dr. Neil Nathan says, our mold expert. If you have a child that does not react well with glutathione, just don't give it. It's making things worse. And so you have to just look at it clinically. I would rather try NAC, see if they tolerate that. If not, maybe measure homocysteine level and then treat with B6 to use your own, but always wonder, why are they reacting like that? What are their genes doing? Can you break down the sulfate from glutathione? If not, if the SUOX gene, for example, is the reason that you don't react great to glutathione, then you need to give thiamine and molybdenum. And so, that's why these genetic tests are very highly individualized. We have to look at everything, how does your child poop? How does it behave with certain medications? What have you tried in the past? What has worked? And then look at these genes and correlate with these symptoms, and then come up with a very customized plan of recommendations. Will a neurotransmitter test show mold toxicity? No, it can suggest dysbiosis, with these high elevations of GABA. The only explanation is dysbiosis, again, because, or at least often it is correlated with dysbiosis because, again, most of the GABA in the periphery is produced in the gut. And let's see. How long should we wait after an antifungal and metal detox protocol, or retest an oat or mycotoxin test to be sure we get the right results? That's a good question. And well, for one, it depends on your resources, of course. But I think if you get a baseline and you have a good history and you follow the child very closely with your practitioner, with your pediatrician, whoever you're treating with, you don't really need any more testing. I don't really push patients into getting more testing, because why? We know already what your blueprints are, what your genetic susceptibilities are, what your weaknesses are, what your neurotransmitters are, what your tendencies are. And the most important thing, once you have all of that information, you just start attacking. And with that, you follow the individual very closely and see how they improve. But there are a lot of other good tests, that you can do. You can do the oat test, like I said, to see if you maybe have, oftentimes when we kill mold, and mold toxins, like I said, the sweat contains a lot of mold toxins. That's why sauna therapy is very effective, but also the urine and the stool, of course. But if we dump a lot, if the liver dumps a lot of these toxins into the bile and then into the gut, these toxins cause possible local inflammation again in the gut, and you can have secondary constipations. And if you don't go poop, then it gets reabsorbed and you create more detox symptoms. But specifically, what I've been seeing is that that secondary yeast infections starting to develop because of this inflammation of the mycotoxin dump from the liver into the gut. So, Dr. Neil Nathan often pre-treats with nystatin before he even initiates more heavier guns like itraconazole, amphotericin B. But oftentimes, most of the time, in fact, he mentions that patients don't even need to get to that point, because you can treat with the binders of the mycotoxins, Saccharomyces boulardii, Lactobacillus cerevisiae, casei, different probiotic strains tackle mold. There's bentonite clay, activated charcoal, humic acid, fulvic acid, NAC, glutathione. All of these are counteracting the effects of mycotoxins. But again, it's a fine balance, and we have to follow the patient very closely. Okay, well, I think that's it. I hope that was helpful. If anybody has any questions, please email me. My email has been floating around in this banner down here for, I don't know how long I've been doing this. What time is it? But please contact me if you have any questions. Come visit my website. New website should be up in about 2 weeks. And I want to thank all of you parents for working so hard for your kids when there's no help from conventional medicine. You are the ones that make the difference in your children's lives. It's been an absolute journey and pleasure to be part of it, and so many of you that I've been involved with. And the biggest thing is never give up. The more you discuss this, the more you brainstorm about the symptoms and the problems and the new issues that come up with your kids, the more success you will have. And I'm convinced that every child can be helped in one way or the other. Every child can be helped, and it's up to us to put our heads together and help them, because who else is going to do it otherwise? And so with that being said, God bless you all, and keep fighting, and I see you at the next webinar. Thank you so much.